Good morning, everyone. My name is Paul Stronsky. I'm a father of a now almost 7-year-old daughter with BPAN, and I want to welcome you to this year's research update session on BPAN. I know many of you from previous conferences and also from the BPAN Facebook group, but I want to extend a special welcome to families with loved ones who've been newly diagnosed and are participating in this conference for the first time. Welcome, and we're very glad that you are here. Now, I'd also like to thank our distinguished panelists who are located across multiple continents. I want to thank them for taking their time out of a Saturday, an early morning for some, evening for others. I also want to thank them for everything that they do to try to find treatments, to try to find answers, and to try to improve our BPAN family members' lives. Today, basically what we're going to do is we're going to have our panelists are going to provide some pre-prepared presentations, and then we're going to open it up to questions, Q&A. You can type your questions into the chat function or raise your hands, and we will get to them towards the end of the session. So thank you for that. And now, without further ado, I would like to introduce all of our panelists. We have the first person we're going to hear from is Professor Paul Lockhart, who is a molecular geneticist and cell biologist. He's the group leader of neurogenetic research at Murdoch Children's Research Institute in Victoria, Australia. He uses modern genomic technologies in his research, including next generation sequencing and stem models to study neurogenetic disease. He received his doctorate in genetics from the University of Melbourne. We also then are going to hear from Dr. Robert Kettler in London. He's group leader and manager of the Translational Research Resource Facility at the University College London. He trained at the Max Planck Institute for Immunology in Freiburg, Germany, and the Massachusetts General Hospital in Boston. At University College, he directs a research team with a main interest in molecular regulation of autophagy and mitogenetic signal transduction. We have Dr. Young-Ah So, who is an early stage investigator who received her doctorate in nutritional sciences at Penn State University. She had a postdoctoral fellowship in the departments of molecular metabolism and nutrition at Harvard School of Public Health. And as a nutritional biochemist, she focuses on understanding the roles of iron and manganese in health and disease. And finally, from Oregon, we have Dr. Penelope Hogarth, who's an associate professor of molecular and medical genetics and neurology at Oregon Health Sciences University in Portland. She completed her medical school and neurology residency at the University of Colorado. She played a key role in finding the BPAN gene and led efforts to establish the BPAN-ready natural history study and biorepository. In addition to her work on NBIA, she has extensive experience with Parkinson's disease, Huntington's disease, and other movement disorders. So without further ado, I'm going to turn it over to our panelists. I understand our first panelist is from Australia, and it is a recorded session. So please, let's begin.
Good morning, everyone. Or it's morning here anyway. My name is Paul Lockhart, and I'd like to thank the organisers of the conference for the opportunity to talk to you a little bit about our research today. A new program, a relatively new program to my group, which is investigating the neurobiology of beta propeller associated neurodegeneration. This work has been performed at the Murdoch Children's Research Institute, which is a private research institute that's part of the Royal Children's Hospital, a purpose-built campus facility in Melbourne, Australia. Today I'd like to give a little bit of information about my background and the background to the BPAN research that's been performed at MCRI, and finally to end up giving you some information about our progress to date and our future plans. So in terms of my background, I grew up in a town called Goodnight, which is literally at the end of the road. You may not be able to see it clearly, but this road here leading into Goodnight is called Lockhart Road, and literally when you get into Goodnight, the road comes to an end. I grew up on this farm here, which you can see just next to the Murray River, on a 10,000 acre, broadacre wheat sheep property, which since that time has been converted into an almond plantation, which is what you can see at the moment on the screen. Science and biology was an ongoing interest for me while I was growing up. and I followed through by undertaking my Bachelor of Science in Genetics at the University of Melbourne and completed a PhD at the University of Melbourne also, with a thesis titled Molecular Analysis of Copper Transport in Sheep. So clearly the farmer could leave the countryside but couldn't get away from his background. I then went on to undertake postgraduate studies at the Mayo Clinic in Florida, USA, where I studied neurogenetics and Parkinson's disease with Professor John Hardy. So at the time when I was there, this was the new research building where I conducted my research in Jacksonville, and the centre was dedicated to using cutting-edge genetics to understand and treat inherited diseases. And from my perspective, a very valuable outcome of my training time at the Mayo has been able to transfer these skills in research when I moved back to Australia. There were 3 life lessons I learnt about Florida in the 4 years that I was there. The first is that it rains a lot. This was one afternoon, started raining for 2 hours and then stopped, and as you can see, the front yard of our house was pretty much underwater. The second thing I learned about Florida is that it is impressively cold. We lived within 100 metres of the beach, and this was a fountain that flowed the entire evening, and when we woke up in the morning it was frozen solid. And the last thing I learned while I lived in Florida was that deep fat fried turkey is actually delicious. So why are we performing BPAN research at MCRI? Well, there are a number of reasons. Firstly, there are a lot of synergies with my previous and existing research, which is focused on Parkinson's disease and another disorder, an NBIA, RAB-mediated NBIA. Secondly, Murdoch Children's Research Institute is the major provider of genetic services to 2 states in Australia, servicing approximately 1/6 of the country's population, and this service includes diagnostic sequencing for childhood genetic conditions. And the third reason why we're performing this research at MCRI was from our interactions with David Hunter, seen here on the right, with his partner Edwina on the left and their son Angus. I'm going to play just a small clip now, which was taken from the ABC, our national broadcaster, which I hope will be informative, giving some background into the project and how it came about.
Hitting the road, searching for answers. The Galash family from the Adelaide Hills hopes to find them in Melbourne, amongst people they've never met.
I think all the families want to know what links us, because we're all scattered in so many different countries. We've got no link at all between us.
That is, except for one crucial thing, BPAN, a rare and random neurological condition which in childhood makes learning to stand or walk a struggle. Then, in early adulthood, those hard-fought gains are lost. Once, Amy Galash could walk and feed herself. There are 13 known cases of BPAN in Australia, many of them in this room today.
There's so little that's known and it's so infrequent.
BPAN was only identified in 2013, with Amy Galash diagnosed a year later. After discovering his son Angus's diagnosis in 2016, David Hunter raised the money to start Australia's first research program.
We're hoping that some of these great minds will come up with a solution, hopefully in time to benefit Angus.
The researchers will take skin and blood samples, growing stem cells, to discover how BPAN does damage. No one is sure what they might find.
We see examples of treatments that can be miraculous. We also see conditions such as Parkinson's disease, where we've been studying what's going on for a very long period of time, and we're not really any closer to a cure.
But already families have discovered something valuable: hope.
My hope would be they'd find something to cure her.
Realistically, it might be beyond our lifetime or beyond Amy's lifetime, but for the sake of any others who come that have the same BPAN.
The first step on a long road. Simon Royal, ABC News.
So hopefully that gives you a bit more of a background into how the research came about, and I'll talk a little bit now about the challenges and joys of research in a pandemic. So as you may have gathered from that slide deck, we started initial discussions with David Hunter and various other individuals back in 2018, and in March of 2019 we were able to secure initial seed funding to initiate the project. Using that seed funding, a postdoc was appointed in September of 2019. And at the beginning of 2020, we were actually lucky enough to be awarded some grant funding through the $1 Million Bike Ride program. Unfortunately, in March of 2020, MCRI research was shut down as a result of the initiation of the pandemic, and indeed Melbourne itself went into its first lockdown in June of 2020. Subsequently, in August of 2020, Victoria, the entire state of Victoria was declared in a state of disaster, and the second lockdown was imposed, which also included a complete curfew from 5 o'clock at night until about 7 o'clock in the morning, which was unprecedented. Thankfully, by November of 2020, that lockdown came to an end, and 1 month later, Victoria was declared virus-free, as we'd had 28 days pass consecutively with no evidence of community spread of the condition. And in February of 2021, MCRI research actually reopened, and so now we are fortunate enough to be able to continue on this much interrupted project. So what is the research plan? What we're hoping to do is to investigate and try and understand BPAN a bit better using patient-derived cells to model BPAN. And I imagine some of you in the audience will have come across the term induced pluripotent stem cells, which are essentially where you take a tissue sample from an individual, generally skin, but can be derived from blood, and using a set of molecular tools we can convert those cells into what's called a pluripotent stem cell, and these cells can be converted to any type of tissue in the body, and that may include cardiac tissue, it may include lung tissue, but in the context of BPAN, what we're particularly interested in is neurological tissue and generating neurons, particularly neurons of the brain, that are thought to be affected by this condition. And what we hope to do by understanding the condition using these cell types and also performing drug screening and discovery research, we hope to be able to identify new drugs that might actually be able to treat the condition. So what's been the progress to date? As I indicated previously, it's been a very interrupted program so far. However, we have managed to accomplish a number of milestones, and the first of which was, as you saw in the news clip, getting a very large number of the known families in Australia together in one room at one time to talk about BPAN and to launch the research. And so from that event, we've managed to recruit 8 families to our research program, including 8 affected individuals, which is 1 male and 7 females. In all of these cases, we've managed to identify the causal mutation and validated it within these probands, and we've shown that the male carries a somatic mosaic variant, meaning that unlike females, where every cell in their body carries 1 copy of the mutant gene, in the male only about 50% of his cells carry that gene mutation. And for each of these 8, we've taken skin samples and we've established IPS, pluripotent stem cell lines, and also at the same time we've managed to correct the mutation within those stem cell lines to generate what's called an isogenic control. And what that means is we are able to compare the mutant cell and the corrected cell side by side to look for treatments that might help improve the phenotype that's associated with that BPAN mutation. And we've since moved on to testing these cells for their ability to form neural networks, because as I said, we're particularly interested in modelling this disorder in neurons, the type of cell within the brain that is most affected. And here you can see some examples of that early work where we've converted these IPS cells into neurons, and they're forming really nice networks where they're connected to each other with these fine string-like structures and are able to communicate with each other. And we're hopeful that this will model to some extent the communication between cells in the brain. So what's next? What are our plans for 2020-21? In the next couple of months, we hope to complete the final characterization and testing of these cell lines and the neurons that we are able to generate from them. In September, we're hoping to perform a compound screen of approximately 4,000 FDA-approved drugs. So essentially, this process involves seeding the neurons into these plates, treating them with these different compounds, and then looking at phenotypic readouts. And what we're hoping to see is that we will be able to correct some of the phenotypes, what's wrong with the cells when the mutation is present. And obviously after that initial screen, we would be looking to identify lead candidates for further research and validation, with the ultimate goal of transitioning those to whole animal studies and future clinical trials. So I'd like to say thank you for your time and interest, and a big thank you to all of the families and supporters in Australia and more broadly who have been associated with this project. Particularly want to thank Professor Martin Dalaticki, a clinician researcher indicated here, who works very closely with me on this project, and also Jay Shukla, who is just hiding here, who is the postdoc doing the hands-on work involved in this process. Particular thanks to the Murdoch Children's Research Institute for all their support, and of course the Orphan Disease Centre at the University of Pennsylvania, who have initiated the $1 Million Bike Ride program, which we were able to successfully gain some research funds from. Thank you very much for your time and interest.
Good morning, everyone.
Yep.
Thank you very much for the kind introduction, and it's an absolute honor and pleasure to present some of our research today. My name is Robin Kettler. I work at University College London, and this is where we do drug screening in iPSC-derived neurons from patients. So first, some of the projects ongoing in the lab. We generally like to develop therapeutic strategies for several diseases. Today, I will talk about our drug screening for BPAN, which is a great collaboration with Professor Manju Kurian at UCL. But we also have programs where we try to develop therapeutic strategies for Parkinson's disease. We do this largely in collaboration with a Japanese pharmaceutical company. And we also have a program where we try to develop inhibitors of autophagy in pancreatic cancer in a collaboration with AstraZeneca. But today I will focus on the BPAN project. And these are the two main aims of the project. We'd like to understand what is different in patient cells and what differences might be contributing to the disease. And then in a second step, we'd like to correct these differences in patient cells using genetic tools, but also small molecule chemical compounds. So first, genetic perspective of BPAN. BPAN is the consequence of a single gene mutation in a gene called WDR45 or WP4. The link of this mutation to disease is not well understood. We know a little bit. For example, we know that this gene functions in a pathway called autophagy. And I will talk a little bit about autophagy during the course of this talk. Now here on this image on the left bottom, you can see how we generally visualize proteins. It's usually very colorful ribbons and loops of the structure. And on the right, you see a scan of a brain. And here you can see the brain iron accumulation, which is one of the disease hallmarks. So autophagy is a very important process. This person here, Yoshinori Ozumi, has been awarded the Nobel Prize in Medicine in 2016 for discovering this process. And he and others were able to show that actually no animal can survive when autophagy is defective. Usually this is during the embryonic stage, but in the adult state, neurons generally die due to toxic buildup of damaged cell material when there's no autophagy. So this is a really important process. So in terms of the cell biology, autophagy is derived from the Greek word for self-eating. And this is literally what happens during autophagy. Cells, when they are under starvation, for example, they start to eat themselves up. And this is a good thing because then they provide additional energy to survive this starvation. Autophagy also operates to remove damaged cell components and toxins from cells. Now, if they are not removed, then this can lead to death of the cell, and this is usually very detrimental in neurons. So on these images here, on the left bottom here, you can see these spots. So this is how we visualize autophagosomes in cells. Each of these spots is where this self-eating or the removal of a damaged material happens. Here on the right is an electron microscope image. So we go even deeper. We show very fine structures. And here we can see, for example, here this cup-shaped double membrane form is starting to engulf this mitochondria. I'm not sure if you can, oh, here's a pointer. Maybe I can do this. No. Anyway, I hope you can see my pointer. And here a damaged mitochondria is taken up by the autophagosome. So now, this is a schematic of the whole process. So initially, a small phagophore forms around the material that should be removed from the cell. Then this round structure closes and ultimately it fuses with the lysosome. And that's where this damaged material is broken down in very small parts, which are then given back to the cell to provide energy. So the autophagosome delivers the damaged material to this lysosome, and the lysosome degrades that material to provide more energy. In BPAN, the gene that is defective operates at this very early step. So it helps to recognize cargo to initiate the formation of the autophagosome. When this is defective, none of these further downstream processes can take place. And the damaged material accumulates and ultimately kills the cells. And this is most damaging in neurons in the brain. So what we like to do is correct the gene defect. We'd like to put back the healthy gene. And we'd also like to restore autophagy by eventually developing small molecule chemical drugs. So this is, these are the entry points where we can operate. We can either try to upregulate the early steps, so the number of autophagosomes. We can try to make this process more efficient and faster, or we can improve the degradation of the toxic material at step 3. But we are mainly focusing on the first step, that is the increase in the number of autophagosomes. So again, our rationale is first to understand what is different in patient cells and what might be contributing to the disease. And then secondly, to correct these differences in patient cells. So we look at cells where WDR45 is mutated. And then we also look at all the other genes that are up or down regulated in these cells. And then secondly, we'd like to use a genomic tool, so CRISPR-Cas9 genome editing to correct the defective gene, and also like to correct all of the defects and autophagy by drug screening. The ideal disease model is really to look in neurons. However, you can all appreciate that's really difficult to study neurons from patients. So therefore, we have started using pluripotent stem cells that we can then reprogram into neurons. And again, this is something that absolutely fascinating technology that has been awarded with the 2012 Nobel Prize in Medicine. Shinyaka Yamanaka developed protocols where any somatic cell, so for example, skin cells can be reprogrammed in stem cells, and from there we can reprogram or differentiate them into any other cell type in the body, including neurons. So this technology then for the first time allowed us to use skin cells from patients and then mature them into neurons and study patients' neurons. And this is the workflow, how we do this. So we get skin fibroblast biopsies, we reprogram them in iPSCs and then differentiate into neurons. We also use CRISPR genome editing to correct the gene defect and then differentiate into neurons. And then we have healthy neurons and neurons with the patient mutation. And then we can look at differences between the two neurons. We can then use them for drug screening and to understand the disease in more detail. These are the fibroblasts from patients and healthy controls that we used. We have 2 control lines, we have 3 patient lines, and then at the bottom, 2 of the patient lines we have corrected by CRISPR-Cas9. The differentiation into neurons from the stem cell takes quite a long time. So it takes us 65 days to grow the neurons in the lab. So we have to be very patient. However, at the end, we can see here with this green marker, which stains the neurons, and then this red marker, which stains a subtype of neurons, the dopaminergic neurons, which are responsible for controlling motor movement. We can see that we have successfully generated the right type of neurons in the dish. So then next, we were interested whether we can see differences between the patient's neurons and the healthy controls. We used RNA sequencing where we can sequence all of the genes in these cells, and that's the whole genome. So all genes in the samples. This heat map here shows in red color all the genes that are upregulated. For example, here in the healthy lines, and in blue color, all of the genes that are downregulated. And we were really surprised. Remember, there's only 1 gene defect in these cells, so WDR45 or WP4. However, we see differences in genes in over 200 genes. So that indicates that this single mutation leads to a lot of changes in the cells, and this is what we try to understand a bit better. We were quite pleased to see that many of these genes that change are involved in iron metabolism, which is one of the hallmarks of the disease. So we think we have a really good cell model here. To correct the phenotype, we are using CRISPR genome editing and drug screening, as I've mentioned before. Just a little bit about CRISPR genome editing. Some of you will have heard that the latest Nobel Prize in chemistry this time, 2020, was awarded for the discovery of this genome editing technology. And these are the two very prestigious scientists, Emmanuel Charpentier and Jennifer Doudna, who made this possible. I have a quite complicated slide here, but just to say how CRISPR works. So this pink strand in the middle here represents, for example, the gene that we'd like to modify. CRISPR makes a cut like a scissor, and then this turquoise element here is a corrected gene. So we can correct the gene defect quite easily and cost effective in this manner. And CRISPR really allows us to do this to any gene at any location in the genome. What we've seen here, so again, a relatively complicated slide. Just look at these blue bars here, which represent, in this case, the WDR45 gene. In the middle, the three patient lines, we see no WDR45, which is the defective gene, but we do see it back in this CRISPR corrected lines, which are here at the end. The two lines have the same level of the gene as the healthy controls. And then on the right here, these bars in yellow in the middle are one gene that is upregulated in the patients, which is ferritin heavy chain in our samples. And this upregulation, which might indicate the brain iron defect, is downregulated in the CRISPR lines. So we have CRISPR lines now where we can correct the gene defect and also some of the iron phenotypes. And then ultimately, we then went on to do drug screening, and this is how we do it. We have a chemical library. We add this to the differentiated neurons, and then we acquire images in a high throughput screening microscope, which is basically a box and then a microscope inside. And this allows us at the push of a button to acquire thousands of images in a very short time frame. We get then images back. We have to analyze them by quite complicated bioinformatic tools. And this will allow us then to count the number of autophagosomes in these cells. This is some of the equipment we're using. My lab has a lot of robotic automation, and we can do these processes in very high throughput, going through thousands of samples in a day with minimal intervention from us researchers. So we set up these machines and they just operate by themselves, more or less. For us, the most important part is then the analysis of the data and making sure that we get good quality data. We have decided to screen an FDA-approved drug library. So these are compounds that are already used in the clinic for other diseases and indications. The advantage is that therefore we know the safety and toxicity profile of these compounds. They are relatively safe to use. We know how they work. And overall, this should lead to a much faster route to clinical approval for BPAN. The disadvantages are it's only a limited number of compounds that are available and a limited number of target genes and pathways. And if we're unlucky, we may not find something that helps for BPAN. This is what we've done so far, just a schematic. So we use 96 well plates. So we have 96 compounds on each plate. We go through several of these plates every day. And at the bottom here, in these images from the microscope, you can see in blue, that's the nucleus of the cells. So you can see the cells are blue. And then in green in the middle, you see that one of the compounds actually increases massively the number of autophagosomes. And this is in a patient line. So we were very pleased to see that we have compounds that can correct the defect in autophagy in these lines. And this is a summary slide of the screen. These are 16 plates, 96 well plates that we've used. And on the right here with this green circle, you can see all of the compounds that increase autophagy. So each dot is one chemical that increases autophagy in a patient's cell line. So we identified several chemicals that can enhance autophagy in the cells. And we are now studying further their effect on other processes, and we aim to develop these into drug candidates. So in summary, we have generated stem cell lines, iPSC lines from patients, and differentiated them into dopaminergic neurons. We have them corrected the gene defect in these cells, and we see some correction of phenotypes. We have observed a defect in autophagy and in iron metabolism in these cells, and screening the FDA-approved compounds has identified some compounds that are very promising for further development. Our next steps will be to do chemistry to improve the compounds because at the moment they are not suitable for any treatment in the patients, unfortunately. And then we will also continue with our data mining to better understand the consequences of WP4 loss. And we will also aim to improve our patients' disease models further, for example, trying to develop three-dimensional brain cell models. And the most important slide, the acknowledgements. This work was mainly done by Apostolos Papandreou, an absolutely brilliant PhD student in the lab. And you can see in this small image here how he chatted with his fellow Greek colleague, friend, Prince Philip, which was 2 years ago, I think. And this whole work was done in collaboration with Professor Manju Kurian at the Institute of Child Health and a very talented postdoc in her lab, Selena Barral. And I'd like to acknowledge all of the funders, including the NBIA Disorders Association, $1 Million Bike Ride, the BPAN Association, and Action Medical Research, which all funded this project. Thank you very much. Looking forward to questions.
All right, thank you so much, for giving me an opportunity to present our work on the NBIA, especially in the BPAN. So as you know, the previous presenters already kind of introduced about what is the BPAN, but I will just briefly summarize it. The BPAN is one of the subtype of NBIA, and then NBIA is a group of a heterogeneous group of the neurodegenerative disease, characterized by abnormal ion accumulation in the brain, and also the progressive extrapyramidal dysfunction. So the most prominent, the ion accumulation happens in the brain, especially those the basal ganglia. As you see those MRI images, and then as well as particularly in the subtype of some of the basal ganglia, which is called the globus pallidus and the substantia nigra. So then WDR45, the gene is kind of a causative genes for the BPAN, and then WDR45 is either the mutated or deleted in the BPAN patient. So what is known about the WDR45? Just name is coming from those amino acid ending with the tryptophan and aspartate. That is a kind of abbreviation for the amino acid WD, And then the structure of the protein is looking like this, 7-bladed, beta propeller. So that's where the BPAN name is coming from. So what is known about the WGR45, which is, known to regulate the autophagy. So I think Dr. Kessler just, talked about, the insight about, what is the autophagy. So I'm not going to talk about it in detail. However, just, to simply speaking, the autophagy is kind of the helping to clear unneeded materials from the cell, including the excess amount of iron, storage protein called ferritin. So there is, a word called, the autophagy plus the ferritin is called the ferritinophagy. But how does WGR45 contribute to the ferritinophagy? It's not known yet. Then WGR45, what is known about autophagy is using the BPAN patient-derived the lymphoblast cells or the mouse models of WGR45. What they do is they specifically deleted this gene, coded gene for the BPAN specifically in the neuron or whole body. And then what they found is, those BPAN patient-derived cells or the mouse model, overall shows, autophagy problems. So impaired autophagy blocks. And then also they have shown those learning and memory defects and then cognitive impairment as well. So those, the mouse models recapitulate what is known in the BPAN phenotype. However, they didn't really show the iron accumulation in the brain yet. So then the next question becomes, how does WGR45 play a role in autophagy, and how does it relate to iron accumulation in BPAN patients? So to talk about the iron accumulation, what is the iron? So it is a metal, but it's biometal, so it is essential. However, it could be toxic to our body. So it is actually very important, essential trace element that we call minerals required for almost all living organisms, from the yeast bacteria to humans, all need iron. Especially for the brain. The brain is the most metabolically active organs in the body, and which requires iron because of the function of the brain, such as neurotransmission or myelination, mitochondrial energy production, oxygen transport, cellular division, and many other the functions of the brain requires iron. So it's very important and essential. However, for some reason, if it is excessive iron is accumulated in the brain, it should be very toxic because of the iron, excess iron can generate what we call reactive free radicals, and then those radicals can attack tissues, and there was the DNA, lipids, proteins, so it causes the tissue damages. So this is where the iron accumulation causes the neurodegeneration as well. So then the main question becomes, let's say WGR45 is known to regulate autophagy, and the WGR45 is known to accumulate the iron in the brain. So then how the roles of the WGR45 in autophagy is related to iron accumulation in BPAN patients? Those questions have not been addressed. So to address those questions, we actually generated WGR45, the deleted cells in the neuroblastoma cell line using the CRISPR-Cas9 mediated genome editing. So this is how we generate the cell line, and then using the ICP-MS, which is the mass spec to detect the iron levels in the cell, we found that here is the wild type, which is a control, and then this is WGR45 knockout cell, which is kind of mimicking the BPAN patient phenotype, and then shows iron level is twofold higher in the WGR45 knockout cells. So which shows that this cell line reproduced what is observed in the BPAN patient. So we next look at what is the mechanism, how the loss of the WGR45 increased total iron level we just observed, right? So to identify those mechanisms, we're looking at the cellular iron uptake pathways. So if you look at these cells, there is two pathways exist to take the iron into the cell. One is through the TBI, which is the transferrin receptor. You can see here, one of the iron receptors, they can accept iron into the cell. And then the other way of taking up the iron into the cell is non-transferrin bound iron uptake. So there are two specific iron uptake pathways. So we are looking at which pathways is important to accumulate the iron in BPAN. So using the radioisotope of iron-59, which is the most sensitive way to detect the iron uptake, and then looking at the kinetics, we examined the uptake of iron-59 into this cell. And then what we found in here is in TBI uptake, there is no difference between the wild type and KO cells. However, in NTBI uptake pathway, as you see here, compared to the wild type, WZ45 KO cells increase more iron uptake by over time. So which means, the NTBI uptake is significantly increased in WZ45 KO cells, which may contribute iron accumulation in the cell. And then we next look at what kind of transporter or iron-related factors can be unregulated or downregulated. So we look at the gene expression level. And as you see here, with the TBI is associated with this transferrin receptor proteins and transferrin, and then NTBI, the transporters related with one is DMT1 and the other one is ZIP8 or Z14. And then also there is an iron exporter protein, which is called ferroportin. So we're looking at all different gene expression levels of these proteins. And as you see here, compared to the other genes, you see the SLC11A2, which is DMT1, the gene name. The SLC11A2, which is DMT1, is highly unregulated in the WZ45 KO cells. So this data suggests that those DMT1 mediated iron uptake is highly unregulated in this WZ45 KO cells. And then we next look at once the iron is accumulated in the cells, where did it go, right? Once the iron is getting into the cell, either it should be used in the cells or it should be stored in the protein called ferritin. So the ferritin is a protein called iron storage protein. It's very important to maintain cellular iron homeostasis. And what we found in here, correlated with iron uptake and an iron, increased iron level in the WZ45, the ferritin expression level, protein expression level is significantly higher in the KO cells compared to the control. So which means, when iron is taken up into the cells, in the WZ45 KO cells, it is stored in the ferritin, and then those ferritin, the expression level is getting higher. So then, I just briefly mentioned when I talked about the iron, there is, iron-related autophagy process, which is called ferritinophagy. So once the iron is, stored in the ferritin has to be sequestered within the autophagosome, and then it is delivered to the lysosome for degradation. So to simply, explain this, once ferritin is, bounding iron, right? And then it has to be degraded and then release the iron to the cells. Otherwise, the ferritin, if the ferritinopathy is impaired, iron is accumulated in the ferritin. And then as you see here, to determine where if the ferritinopathy is impaired in this WZ45 KO cells, so we did some experiments. And then as you see here, without any treatment, compared to the wild type, the KO cells has increased the ferritin level, right? However, when we treat with the DF4, which is the iron chelator, to stimulate the ferritin degradation, we found that the wild type, as you see, the DF4 treatment induced the ferritin degradation. However, the DF4 treatment in WZ45 did not induce the ferritin degradation. And then also the ferritin deficiency has to be dependent on the NCoA4, but we did not see that any changes in NCoA4. So this impaired ferritin deficiency is independent of NCoA4. So what is this data really show us is WZ45 KO cells has impaired ferritin degradation. So once iron is taken up into the cell more compared to the control, it is stored in the ferritin. But in natural process of iron metabolism, ferritin has to be degraded. However, in the KO cells of the WZ45, those ferritin degradation does not happen, so all the iron gets stuck each other in the ferritin. So because of limited time and space today, I'm not going to show all the data we produced. However, what is the summary of what we found in this cell line is compared to the control, in WZ45 KO cells, we found that through the NBIA uptake, the DMT1 mediated NBIA uptake is significantly increased in WZ45 KO cells. And then there is more iron is stored in the ferritin. So then once the ferritin is stored in iron, they have to be degraded at some point, and then so release some free iron outside of the cell. However, in the process of the autophagy, we found that the lysosomal function is impaired in WZ45 KO cells. That's why the ferritinopathy is all impaired. And then at the same time, we also found that the iron the taken up into the cells are all highly accumulated in the mitochondria. The mitochondria requires iron. However, too much iron in the mitochondria will cause some problems, which is called kind of apoptosis here. So based on this study, what we really found in here is WZ45 actually linked to specific alteration of the iron acquisition pathway, uptake pathway, and then lead to the impaired ferritinopathy, and then also neurodegeneration. So then based on the mechanisms we identified here, we are currently working on the developing therapeutic targets. One way of looking at those therapeutic target is this we can inhibit the DMT1 mediated iron uptake. And then the other way of to fix this is to rescue those impaired the ferritinopathy. One way of to looking at those impaired ferritinopathy rescue is kind of we try to reduce the ferritin level in the cell. So the study is still going on, but one of the manuscripts is still under review, and then it will be probably available in several months. And then lastly, I really like to thank to who does most of the work by the Louisa Aring, which is the doctoral student in my group, and then also the funding source of the NBIA. And then I will be happy to take a question at the end of the session.
So I'm Penny Hogarth. Thank you to the organizers for inviting me to talk today about the BPAN Ready study. And the real truth is I'm actually not going to do most of the talking. My colleagues, Katrina Wakeman and Dolly Zen from the Hayflick Hogarth Lab will be doing a good part of this presentation. So I hope it all goes smoothly. However, I am going to start with a story to kind of tee this up. So many years ago, I met a family. Just see if I can get this to advance properly. Well, that's a little bit of a problem. There we go. So many years ago, I met a family. It was actually at one of the NBIA Disorders Association meetings. Totally lovely family. I think they might be involved in this meeting even now. They'd found their way to this community because their son had been found to have, their adult son, found to have iron on brain imaging. And his story was very different than most of the other people, all of the other people at that meeting. He had had intellectual disability in childhood. He had very limited language development. And then in early adulthood, he started to have trouble with gait and balance. He was found to be Parkinsonian. He looked like he had sort of an odd form of Parkinson's disease. And that prompted some imaging, which revealed the brain iron, and he found his way to our community. And then just a year or so after that, we met another family in clinic with a very similar story for their adult daughter. That prompted us to wonder whether we had another genetic disorder that hadn't been identified, that we needed to go searching for, and really in work that was spearheaded by Alison Gregory in our group. We mined the data in our, by this time, substantial registry. And we found 13 patients who had a very similar story. They were all adults and they all had this same kind of story. And in addition, many of them had seizures in childhood. And that ultimately led to a collaborative project with a researcher in Germany. It was funded by the NBIA Disorders Association. And that ultimately led to the discovery of the BPAN gene. And what's followed is just a grand example of all the things that Dr. Eric Sid talked about yesterday. We quickly developed a gene test that could be used for diagnosis, and we began to think about going beyond our registry to develop a natural history study and dedicated biosample repository. And we knew that this would aid in our understanding of the development of the biology and progression of BPAN. It would guide families and clinicians in care, and it would ultimately serve the design of clinical trials and the development of treatments. This is exactly what Dr. Sid was talking about in his presentation yesterday. We held our first meeting of just about 20 BPAN families, and it's hard to believe, but this was only 5 or 6 years ago. We held focus group meetings there, and that guided the development of the natural history study. And the study has already informed the development of the best practices guideline, which will be published and available to families and clinicians just in the next couple of months. This is work that's been led by Jenny Wilson in our group at OHSU. So today we're going to give you a very quick snapshot of the study, and I would emphasize snapshot though, and point out that the real power of this study is going to come from data that we're collecting very rapidly over time from your children. The more people that we can enroll in a natural history study, the earlier in life, the better. The more time points we collect, and the more power the study has to detect change and to reveal things about BPAN. And as Katrina will show you, the BPAN family community has amazingly risen to this challenge. You've not only been willing to enroll your children in impressive numbers, but most importantly, all of you have stuck with us. And I'd just like to give a particular call out to Alison Gregory, who really helped design the study, has spearheaded much of the work on this. I play a bit part here. So with that, I'm going to hand this over to Katrina. I'm going to run the slides, and I think Katrina will tell me when to advance. So Katrina, I'll have you unmute and take it away.
All right. Hi, everyone. My name is Katrina Wakeman. I'm the study coordinator for BPAN Ready. I think many of you have heard from me, as I'm the one calling you and emailing you every 6 months when it's time to do visits. So I'm going to go over the study. You can see here kind of the goals of the study. As Dr. Hogarth talked about, many of the first people that we found with BPAN were older, and so we wanted to get an idea of what BPAN looked like in younger children as well as adults. And then we also want a study where we can try and find a symptom or a measurement that can work to kind of track the disease, see changes and progression, and then be used to measure if future treatments are working. Next slide. And then we have 5 general focuses in the natural history study, and these were all derived from some original focus groups with people who have BPAN or their children have BPAN. So we ask questions about seizures, motor delay, language delay, behavioral issues, and sleep disorders. Next slide. Okay, so when I'm calling all of you. I get a lot of questions about how many people are in BPAN Ready, and then, what issues other kids are having. So, my kid is dealing with XYZ. Does other people in the study deal with this too? So I wanted to go over just a really small snapshot of the data to kind of get at these questions. Next slide. So currently we have 115 people active in the study. We had 120, but some people drop out. And then you can see that the vast majority are female, and that makes a lot of sense since it's an X-linked disease. Next slide. And then this is a pie graph that shows the different age ranges in BPAN Ready. And what you can see here is that over half of everyone in the study is 10 years or younger. And this is really helpful for us because, like we said earlier, we had kind of a better understanding of BPAN from older people. And so this has really allowed us to see what BPAN looks like in younger groups and kind of the range that there is for BPAN. And then you can see that we also have, about 25 to 30% who are 16 years or older. And that's really helpful for knowing kind of the different set of problems that older people with BPAN are going through. Next slide. And then here you can see just the general demographics of the study. So there's the ancestry of everyone in the study, and then whether or not people are Hispanic or Latino. Next slide. And this slide shows all the different countries that people in the study are from. You can see that the majority are from English-speaking countries, and that is because at the moment we don't offer the questionnaires in other languages. And part of that reason is because we use validated questionnaires, and those questionnaires are probably not validated in other languages. And it's a bigger task.
Katrina,
what was that?
out a little.
Okay. Can you hear me now?
Sorry. Katrina, out a little bit, rather.
Yeah, we can hear you, Katrina.
Okay, go ahead.
Yeah, we can hear you.
Go ahead. Okay, great. Sorry.
Sorry, Katrina. Go ahead.
That's okay. So as I was saying, yeah, so the questionnaires are validated, and that's a bigger task to translate those. It's not simply changing it to another language. And we would really like to do that in the future, probably starting with Spanish, but it's something that we don't necessarily have the resources or funding for at the moment, but is a long-term goal. Next slide. And this just shows that 29 of 50 states are represented, and that's just because we've worked really hard to have all our natural history studies be able to be done remotely so that we can have wide participation. Next slide. Okay, and this is a bit of a confusing graph, but I'm going to do my best to explain it. So you can see that this is showing study progression and how many visits have been completed and which visits have been completed. So the most number of people have completed visit 1, and then the least number have completed visit 6. And what this means is that those who have completed visit 6 are the people that enrolled right as the study started. So they've been with us for the last 3 years, and that's why they're furthest along. And visit 1, there may be some people in that column who just enrolled a couple months ago, and then everywhere in between for the other visits. And so what that means is we've had really good enrollment over time. It's not that we had a big chunk of people come in at the beginning and then no one's enrolled since, but it's been quite the opposite of steady enrollment over the years. And you can see that there's a total of 443 visits completed to date. And like Dr. Hogarth said, some of the most important part of this study is getting data over time. So we want to have everyone in the study keep completing their visits beyond visit 6 so we can see in real time how BPAN changes over time. And then at the same time, we'll be working, and other researchers are working to develop therapies so that we can do that in parallel with the natural history study. But the more data we can get in the natural history study, the more ready we'll be when something is ready to go to clinical trial. Next slide. So now I wanted to kind of go through some of the common symptoms that we saw through the focus group and that have come through, have been shown through BPAN Ready. And the first one I have here is disturbed sleep. So this represents anyone who has trouble sleeping through the night regularly, wakes up 2 or more times a night, or takes more than 30 minutes to get back to sleep. And you can see that 62% of participants in the study have had disturbed sleep at one point in their lives. This doesn't mean that they all currently have disturbed sleep or that they've had disturbed sleep their entire lives, but it's just at one point they were struggling with sleeping through the night. And that's true of all the data I'm going to show you, since it is a snapshot in time. And then the next one you can see is seizures. And it's, overwhelming amount have had seizures. 97% have had at least one seizure in their life, but there is a really big range. So some kids may have had a febrile seizure at age 2, while others are having daily seizures for most of their lives. And then the other interesting thing to note is that there are 3% who've never had seizures. So kind of begs to question what's different there. Why aren't they developing seizures, or has it just not been picked up? And then the third bar you can see is mental health or behavioral issues. And about half of people in our study have had mental health or behavioral issues. And then about a little over a third of those have taken medication for this. Next slide. And then I wanted to dive into language development separately, since we do collect a lot of data on this. And here you can see different levels of ability for communication. And with 100% of people in the study being able to laugh out loud, 99% being able to vocalize sounds, about 80% being able to babble or jabber, which is making kind of sounds that sound like conversation but don't have actual words in them. You can see 73% say dada or mama, but only about 61% say it with meaning or say it appropriately. And then a little over a third are able to use at least 2-word phrases. And so you can see that this is participants age 0 to 15. So that means that some of the kids represented in this data are maybe 1 or 2 years old, and they'll go on to develop more language, but just haven't yet. And then other kids are a bit older, and they're kind of at the level that they'll stay at. And we do have more questionnaires and more data on language for those who do speak more than 2-word phrases. I just haven't shown those here. And next slide. Okay, and then the other big issue with BPAN is kind of motor symptoms and mobility issues. So this shows what assistive devices people are using in the study. You can see 58% use leg braces, 29% use walkers, and 45% use wheelchairs. You might notice that that adds up to more than 100%, and that is just because some people are using more than one device. So some people might use leg braces and a wheelchair, or leg braces and a walker. And then about a third don't use any devices at all. Next slide. And then the last part of BPAN Ready is the blood samples that we get from you. So this is where I send you a kit. It has a little blood tube in it. You take your child in to get blood drawn and then ship that back to me. So we actually have 110 of those blood samples that we use for DNA, RNA, and lymphoblast cell lines. We've gotten 80 of which through BPAN Ready. And then the rest of the blood samples and the urine samples and the fibroblast cell lines we've gotten through clinic visits or at meetings like this one in the past when we were in person. And with those samples, we use them for research in our own lab, and we also share those readily with researchers across the world. And I'm actually going to pass this on to Dolly to talk about what we are doing with the blood samples that we have. Next.
Hi, everyone. This is Dolly here. I'm a research assistant for Dr. Hayflick and Dr. Hogarth. And unlike with Katrina, you don't get a lot of emails or phone calls with me. And that's because I'm on the research lab side of things. But I wanted to go over some of the initial data. So Katrina gets all the blood samples and I take them and process them And I end up storing them for either experiments or sharing them with other people. So can I get to the next slide? So I have one small graph here on the right, and I'm gonna just take you through it because it looks a little messy. But on the y-axis here is the biomarker level, which is something we found in the blood samples. And from bottom to top is increasing biomarker level. And on the x-axis here is BPAN symptom measurement, and from left to right, it's more severe to less severe. And from the initial data that we have here, we found that people with lower biomarker level has associated with more severe BPAN symptoms, and with higher BPAN or biomarker levels, we've seen less BPAN symptom measurements. And the data is at the beginning stages, so there's a lot of details being left out here, but they are statistically significant. Because it's at the beginning stages, so we are working to build on the data set with all the blood samples that you guys have sent. And I just wanted to take the time to say thank you for all the blood tubes that you guys have sent and the time you guys do to complete the BPAN Ready surveys. They're very important, and thank you, and keep up the good work. And with that, I'm going to hand it back to Penny.
So I'm just going to say a very brief word about the longer term plans for data analysis. So as I emphasized, this is a snapshot. It's a sort of cross-sectional quick look at the data to give you an idea of what we are collecting. But the real power is going to come in looking at data over time. And there are some challenges in BPAN in particular, because we are needing to analyze data that is coming in, which reflects sort of the beneficial effects of normal development in a child, and what effects the WDR45 gene is having, the negative effects that the gene mutation is having that are impacting development, normal development. That is a tricky thing to do from an analysis point of view. And we've elected to use this complex and quite powerful statistical approach called latent growth curve modeling. It allows us to look at multiple parameters that are changing over time and to see how different factors or variables affect those curves. And in theory, it would allow us to look not just at the population level, but to, in theory, be able to look at individual groups that fall in a particular category. So maybe those with more severe BPAN, those with less severe BPAN by some measure. We could then look to see how their curves go. We could even, in theory, look at the individual child level to derive or develop a curve for that particular child, so that when a therapeutic comes along, we can see for that child whether the curve is changing. It's something that we've done a preliminary feasibility analysis and work that was funded by the $1 Million Bike Ride, i.e. you. And it looks to be feasible, the approach that we're taking. And now that we are starting to collect more data over time, we plan to do another analysis in this coming year to take a better look at that. So I just want to point out this work, the latent growth curve work, is done with some group of very talented psychometricians and biostatisticians at Washington State University and here at OHSU as well. So this is our funders. These are our funders, including the NBIA Disorders Association and the $1 Million Bike Ride. We also had some support from the Clinical and Translational Research Institute at OHSU. So many thanks to everyone. Here's another way you can get questions to us in addition to the session at the end here. And with that, I will wrap it up.
Okay. Well, I want to thank all, sorry for not turning on my video. I want to thank all of our speakers for really very informative discussions. I certainly learned a lot. We've had a couple questions that have come in. I know several of them have been answered already. I've sort of been cutting and pasting those that have been answered. So if you've asked a question, please just go short, make sure that whether it's been answered or not. a few of them have been about, how are you sharing all of your information between yourselves, particularly, you're working across borders. How is that going about? And then also we have a few questions about, how people can, stay on top of your developments, particularly any sort of, I think we have a question from Latvia about, how do they know, when there is a new treatment that might become available? What's the best way to sort of link in from a patient perspective? And I see that Meg has a question as well. Meg, do you want to go?
Yes, I just have to remember how to unmute. So we were diagnosed about 8 years ago, and I was just reflecting on how different the conversation sounds now than it did back then. We know so much more about this disease. I think just a really broad question for all of us families is, the science looks amazing. What does it mean for our children and their lifetime? Can we expect this science to advance enough that it may help our kids?
Boy, Meg, you love to start with the easy ones, don't you?
I'm really good at that.
Well, I'll take a crack. I think the things that are on the positive side of, yes, today's science will have an impact on your child's life, is the fact that we've identified the gene, we have gotten to a natural history study, we have biosamples being shared around the world, the autophagy work advanced to a level. I would just point out that the Nobel Prize for the work in autophagy, a key researcher who led the work in autophagy, that actually happened the same year almost as the gene discovery. And that could have been happening 10 years from now. So that's a good thing. There's a ton of researchers working on autophagy. The work of that was highlighted by Dr. Doudna and Charpentier, I got the name wrong, but Charpentier in France, to look at CRISPR-Cas9, I think is incredibly promising for all genetic diseases. And the work for high throughput screening, I think these things are coalescing, that there is a real possibility that rational therapeutics, so therapeutics that are directed towards the core biology of the disease, are in fact going to make a difference. I think that the challenges are, and I might let Susan chime in here, I think she's lurking somewhere, but getting gene therapy from the lab to the clinic is an incredibly long and challenging process. And I know that there's nowhere, no community where it's felt more profoundly the urgency of needing to move quickly. This is moving incredibly quickly, and I'm astonished at every turn by how fast things are moving from the bench to the bedside nowadays, particularly on the gene therapy front. 5 years ago, I don't think we would have expected that we would be sticking gene therapy into people's eyes and curing certain types of blindness, genetic forms of blindness. And that is actually happening now. So is it possible? Yeah, I think it's very possible. Is it likely? Not sure I'm going to put a number on that. But I think there's every reason to be hopeful. And, Dr. Kettler, since you're live, I will see if you have anything to add to that in particular, since you're doing the high throughput screening work.
Yeah, thanks, Penny. I think I would agree with what you've just said. I mean, the compounds, in an ideal world, we would have discovered compounds that are, FDA approved, able to cross the blood-brain barrier, correct all the phenotypes that we're looking for. Unfortunately, that's not the case. Many of these will require a lot of additional work. But I would say, I think it's really promising that we can correct such a fundamental
And to the question about how do we share, so it's sort of a poignant moment to reflect on the fact that at that very first BPAN meeting that we held, pretty soon after the gene discovery at OHSU, with just 20 families, really, that is most of who was known in the US, and some who traveled from beyond the US, came to OHSU in, I think it was 2015 or 16. And Dr. Papandreou from London, who works very closely with Dr. Kettler, he was the one meeting Prince Philip in that picture. He came to Oregon for that meeting, and in the space of 3 days, he got this crash course on BPAN, and it would have taken him a long time, I think, to really have that intense of an understanding of the disease. It motivated him, I think, to remain in the field. I think he is an incredibly talented guy, and we at that meeting obtained very many skin biopsies. Thank you, people. It was not easy. Skin biopsies, other types of blood samples, and those samples ultimately wound up, many of them went to London and have become part of this work that is going on now. So, it's kind of a beautiful illustration of how you don't know quite what you're going to do with the data when you collect it, but it has payback. We are already seeing those cells from your children being used for high throughput screening assays around the world. And similar work's going on in Australia. And in Europe, continental Europe, there are a lot of constraints on people being able to do biopsies for research purposes on children. And so I think our biorepository has been incredibly helpful for researchers in Europe as well. And there's very important work going on there too.
Great, that's really great to hear. And I think all of us here are very eager to continue help supporting all of your research all around the world in any way that we can. Let me see. I know that I don't know if Sarah Chisholm is there. I know she's had about 3 or 4 questions, so I don't know if she wants to pop on real quick to ask a few of them. We only have a few minutes left. And I will go skim through. If she's not here, I will also go skim through to see what other questions we have not been answered. Great.
What is it we are looking for, Paul?
I was just, well, Sarah Chisholm had a whole bunch of questions, and if nobody's in there, we can also just, they were specific to specific researchers, but she also can probably, oh, she's
okay,
okay,
I gave
Sarah
a microphone. So Sarah, if you have questions, you can turn your microphone on.
So she's got, just a few specific questions. Let me go through them very quickly. She asked for Dr. So, she wanted to know about the lesions of the brain. Was her team able to identify the cause of the lesions of the brain that she sees in her study? Was it due to an injury, vascular injury, impaired blood supply, or due to another identifiable cause? And were these lesions permanent? And also for all the panelists, could they explain the uptick of iron in the brain as affected by a systemic iron status, as can be measured and quantified?
Probably
so I would. Yeah, please, Dr. So, please.
Right. So about the cause of the lesion of the brain, because we only looking at in the cell, the level. What we found is, when iron is accumulated in the cell, it actually induced the free radicals, and then because of that, it caused a lot of oxidative stress, to the cell, and then it caused the cell death. That's what we found, and then it looks like it's not really transient. It was, kind of, keep, occurring in the cell. So probably other like vascular injury or impaired supply of blood, those things could be kind of looking at in the more animal model, but what we found in the cell level was oxidative stress caused by ion accumulation. And then the other, the question from the Sarah, how uptake of the ion in the brain is affected by systemic ion status. So what I understand about the NBIA patient is their systemic ion levels are quite normal. So from the blood work, the ion level is not that high. But what is known is from the mRNA analysis, those brain ion is accumulated in the certain area, which is called the basal ganglia, including those. But I think so what is known is the ion uptake in the brain is separate from what is known in the systemic ion metabolism. So systemic ion metabolism means in the blood work, you wouldn't see the high ion level, but in the brain you will see the high ion accumulation. That's what I have understand. And what is known in the brain ion metabolism is the brain ion homeostasis itself is actually locally regulated in the brain. That's what I kind of understand.
Okay. All right. Any other from any of our panelists on that question, particularly the second one? And then, I think another one, I think this was answered, but this is a question that I think a lot of us have, so I might, bring it up as well. We've heard in the past that, sort of the BPAN affects every single cell in the body. But today we're, talking about stem cell opportunities and CRISPR and all of that, and sort of how does that, it's a lot of our understanding is that some of this stem cell research and just the fact that it's in all of the cells, makes this a little bit more complicated. These new gene therapies. So if somebody could briefly sort of discuss that. How is this applicable, given some of what we hear about it being a whole brain disorder or a whole cell disorder, every cell disorder?
Maybe I can say
Great.
something. So it's really, there's a difference between gene therapy and cell therapy, right? So the cell therapy will be almost impossible because you'd have to change all of the brain cells more or less. But gene therapy is where we just try to correct the gene. So now that might be a better route to do this. And there's some examples where this seems to work well in other diseases.
And we're doing
just to clarify. So we are just using the stem cells as a research model.
Okay.
We don't have the aim to feed them back to patients.
Okay. Anyone else?
Could I just make a comment, Paul?
Please.
So you're exactly right that a lot more types of cells in the brain than just the nerves are affected by this condition. But from the perspective of the stem cells, at least in our research, we're looking at neurons specifically to start with because it's an easier model. And we want to do the best we can in a simple model to start with and then make it more complicated. So certainly one of the things we're interested in doing down the track is to generate astrocytes as well as glial cells, as well as neurons, and mix them together and then do the same screens again once we've got some targeted lead compounds. And hopefully that will be a more robust model of what's going on in the brain. So you're exactly right. We're using very simple models at the start and we'll scale up to more complicated as we develop our understanding.
That's great.
Can I make a comment about,
sure.
And there's also been quite a discussion in the chat about the compounds. People want to know what type of compounds we're looking at and so on. I mean, I would just say it's very early days. So if you read somewhere, I don't know, this and this compound has been identified to correct the phenotype in stem cells, there's a long way to going from that stem cell model in the dish to the patients. And you know, for example, autophagy, I think Matt commented about the magic wand and what will happen if we had this miracle cure. We don't really know because we'd have to, if we upregulate autophagy too much, there may be negative side effects. So we have to titrate exactly how much compound to give and so on. So those are the challenges that we are facing. And don't just buy somewhere on the internet a compound that you may have read about, because that could lead to serious problems.
It's a good reminder as we're all, very eager to find something that will help our children. With that, I know that our time is up, and I know that people need this Zoom space for the next seminar. So I would just like to thank all of our speakers. There were more than 4 because we had several from OHSU, and I think we learned a tremendous amount from them. We want to thank everyone today for their time, all of our experts for their time, and as again for all of the work that they're doing to try to find treatments for our children. And I want to thank everybody for a very good session, and look forward to seeing you on later in the day. Thank you very much, everyone.